I met Dr. Cash, it must have been 3-4 years ago. And actually, you're not a doctor, are you? No, yes. But he's working on it. Now, he's the CEO of Terra Biologicals. And, one of the conferences a few years ago, I got into a great conversation with him and some of the research that he's been doing with respect to oxaloacetate, which, if you go way back into the history of your education, you'll remember is in the Krebs cycle. And I thought, well, that's kind of interesting. Some guy is doing research on one item in the Krebs cycle. And then after talking to him, I realized this is really amazing, the kind of stuff that he's looking at through this one little aspect of the Krebs cycle. It has a lot of implications. I have actually used this product in a few patients with rather dramatic results. One of them was a Parkinson's patient. And I think you're going to talk about Alzheimer's and Parkinson's and some of the latest research you're doing. Yeah. So this is going to be some amazing stuff. So I'm glad you guys have a chance to listen to Alan here. So take it away. You have you got that? You got everything going?
Not sure. Am I on? Can you hear me?
Yeah, you'll be good. Okay.
Thanks, Frank. I want to first thank Frank for putting this together and allowing me to come and talk to you. Frank likes the miracle stories, and as it turns out, I'm one of the miracle stories. In the talk yesterday about trigeminal neuralgia, I had something similar, although it was not in the trigeminal nerve, it was in the glossopharyngeal nerve. Do you know how sensitive your neck is? There's a reason they call it necking. I had an artery that impinged on a nerve, and as my heart beat, it sawed through the myelin sheath and into the nerve bundle. And what that did is it turned on all the nerves at the same time. So it was kind of like putting an electrical appliance to your neck and turning it on. On a pain scale of 0 to 10, it was a solid 10. It was rather horrible. So this happened very, very quickly. It happened one day. I was on the floor. I thought I had had a heart attack. I didn't know what it was. My wife was very clever. She arranged 10 doctor's appointments in 11 days by just calling up and saying, I'll take your first cancellation. I'll take your first cancellation. So I saw a heart specialist. I saw an ear, nose, throat guy. I saw a neurologist. I saw a gastroenterologist. I saw a family practice doctor. I went to an acupuncturist. I went everywhere I could possibly get in 11 days. And it turned out the neurologist said, you know, I've never seen this before, but I've heard about it because it's pretty spectacular. And it's always great to win the lottery, right? And he said, we're going to do an MRI, and if you don't have brain cancer and you don't have multiple sclerosis, then this is what it probably is. So based with those three options, I went and had my MRI. They didn't see anything. And they said, okay, this is what it is. So he gave me some drugs to slow down the rate of pain, but the drugs left me feeling drugged. I felt like I was drunk all the time, and the pain kept escalating as this artery kept cutting in deeper and deeper. So it turns out there are two centers that specialize in this. One's at the University of Pittsburgh, and I was about ready to get on a plane and just go and hang out in their office. The other one was 2 miles away from my house in San Diego, at the University of California, San Diego. So we find this out. My wife calls the guy. I can't even talk at this point because talking is a trigger point. So every time I open my mouth, and she calls head of neurology, who takes her call. That's a miracle in itself. And he says, oh, that sounds terrible. Come in tomorrow morning. So I go in tomorrow, the next day, and give him my MRI. He asked me 3 questions. He goes, well, what we're going to do is we're going to go into the back of your brain here. We're going to find where that cross is, and we're going to pull it apart, and we're going to put in a piece of Teflon. It's like, okay, at this point, I'm ready to have a head transplant. So I know the desperation that people in pain can feel. I was desperate. So he said, well, I can't get the emergency room nurses, the operation nurses that I want because tomorrow's Saturday, but I'll rearrange the hospital's operation schedule and we'll put you in Monday morning. And here are a bunch of drugs. Take as many as you want. Just get through the weekend. Went, did a 6-hour brain surgery. I've been 100% pain-free for over 10 years. And that's because they went to the cause of the disease. How often in medicine do we treat the symptoms? If you treat the cause, you get rid of the problem. So here the cause, in my case, was actually aging. As we get older, our blood vessels, our veins, our arteries get longer, about 15% longer. And while we don't get 15% longer, well, maybe this way, they've got to have somewhere to go. So they fold over, and usually it's no problem. 1 in a million people get glossopharyngeal neuralgia. Okay? It's just bad luck. But the real problem was aging. So I decided, as a physicist, I'd start looking at aging. And what I found when I looked through the literature, that this is what physicists do. We look at lots and lots of stuff, and we try to come up with something really simple, like E equals mc squared or F equals ma. Think of how much power is in that little simple idea. Well, when I looked at aging, the thing that stood out was calorie restriction. We've been doing it for 70 years. It seems to work. So let me start my talk with that. Okay, so I've got 4 parts here. First part, I'm going to go through the mechanism of action of what we've discovered. The second part, we're going to look at current applications as a nutritional supplement, because this product is on the market as a nutritional supplement under DSHEA. We've submitted all our information to the FDA as a new dietary ingredient, which I'm not sure we had to do, but we did regardless. And then we're going to start looking at a future for this compound, potentially as a drug. So we'll talk about some disease. But it's important to separate the two, because we actually manufacture this. And if I talk about this as a disease that you can go out and buy right now, I will be shut down. Okay, so understand that the disease applications are all in the future, if we get approval, and perhaps you guys can help me with that. And the last part, some patient profiles that may benefit from this supplementation. So first, mechanism of action. We've published some of this information in Aging Cell, Open Longevity Science, and other publications. And what we found is that oxaloacetate works through a pathway that is also activated in calorie restriction, specifically activating AMP, protein activated kinase, or AMPK, and using that activation of AMPK to work with transcription factors to turn on and turn off genes. Okay, so your DNA doesn't change, but what does change is the interaction of your genes with environment. And AMPK is one of those modulators that senses the environment through the NAD to NADH ratio and turns on different genes. So picture a seesaw with NAD, which is nicotinamide adenine dinucleotide, which is why we call it NAD, on one side, and NADH on the other side. And these two compounds basically transfer electrons in the cell. They're part of the energy metabolism, okay? And on the seesaw, you've got one side, and at the fulcrum is AMPK that is sensing which way the seesaw goes. And if it goes one way, it works with these transcription factors and turns on and off these genes. And if it goes the other way, it works with these transcription factors and turns on and off different genes. It makes sense. If you're eating a lot, your body has different needs than when you're starving. Makes sense. So, what are some of the benefits of calorie restriction? Why are we interested in calorie restriction? Well, one, it increases average and maximal lifespan by up to 40% in mammals. There are very few things that do that. People have been trying since the age of the pyramids to find things that would do that. Calorie restriction is one thing that throughout the animal kingdom, with some exceptions, increases lifespan. For instance, we don't know if lobsters live longer with calorie restriction because lobsters live several hundred years. We don't know if people live longer with calorie restriction, but certainly we suspect that they will. Some of the other things about calorie restriction is not just lifespan. If you live longer but you're miserable, it doesn't really help. Trust me, I know. Okay? So some of the other things is a decrease in, for instance, cancer incidence. Calorie restriction is one of the best ways to reduce cancer incidence by about 55% as measured in test animals. It's also very effective in neurodegenerative diseases such as Parkinson's and Alzheimer's disease in animal models. Okay? In primates, it eliminates type 2 diabetes. Okay? Does any of your patients have diabetes or cancer or Alzheimer's or Parkinson? Raise your hand if any of them do. Let's see, 1, 2, 99, 100. Okay, so why aren't all our patients on calorie restriction? Simple question. I did it for a month because I study this. My wife suggested that I was irritable. Now we know this is not true. It's an evil rumor. So we started looking at the molecular basis of calorie restriction. How can we mimic calorie restriction and still be able to eat? How can we have our cake and eat it too? To misquote a famous person. So what we found is that the pathway, at least one of the pathways, and it turns out there are several pathways in calorie restriction that can increase lifespan. But one of the ones that's best known at this point is increasing the NAD to NADH ratio. This was first discovered at MIT in 2003 in yeast experiments, little single-celled animals. Okay? Since then, it's been confirmed in a variety of studies. And interestingly, even in human cells, you've heard of the Hayflick limit, okay, where the cells will divide and divide and divide and divide and they'll stop. If you increase the NAD salvage pathway, the Hayflick limit all of a sudden is no longer a limit. Isn't that interesting? Okay? NAD precursors increase lifespan. NAD to NADH ratio activates AMPK, and AMPK activation increases lifespan. Mitochondrial NAD synthesis increases cell survival. I mean, paper after paper after paper. I've got 2 papers that I've got that they came in after this slide was prepared. This is Young, and this is in BMC Cancer. And he's from the University of Leeds. And what he found is that NAD plus levels dictate colorectal cancer liver metastasis. Is that important in living longer, living healthier? Well, if you have colorectal cancer, it probably is, yeah. Here's another one. This is out of Scripps Institute, which is right next to me. This was just published in the Journal of Clinical Investigation. And let me give you a quote from the, they were looking at breast cancer metastasis, so aggressive breast cancer. The quote here is, I have to take off my glasses. Therapeutic normalization of the NAD-NADH balance can inhibit metastasis and prevent disease progression. Is that important? Have you heard about this in the local news? What we need is a good-looking Australian guy who talks about wine instead to talk about this. So, snicker, snicker, snicker. Anyway, so NAD to NADH ratio seems to be an important thing. So how do we get there? Well, there are three ways we've identified. One is calorie restriction. It works. The second one is prolonged exercise. Has anyone heard of this woman who was diagnosed with metastatic breast cancer and she went out and ran marathons? She was given 2 months to live and she ran marathons and she ran marathons and she kept living. She was alive for another 10 years plus. Okay? Increasing the NAD to NADH ratio with prolonged exercise through gluconeogenesis. Look at the charts. You can follow it along. The third way is what we came up with, which was supplementation with oxaloacetic acid. I can't even say it. And what we do is we take oxaloacetate, we react it through the enzyme malate dehydrogenase, which is in just about everything. It makes malate, which is another metabolite. So these are just metabolites. They're in every cell of your body. And it takes NADH and makes NAD, which increases the ratio. Now, a guy named Krebs, you may have heard of him. I had even heard of him, and I'm a physicist. He measured the increase in the NAD to NADH ratio using oxaloacetate as 900% in 2 minutes. Does that change the local metabolic environment? Yeah. Yeah. So what we're doing and what we found is that increasing NAD or decreasing NADH, and there's a great paper on that by people named Raphael Fail, activates AMPK. And then AMPK works with a transcription factor, a FOXO family transcription factor, to actually increase lifespan. Now, if we disable AMPK or disable the DAF16 in this case, which is for worms, we don't see this increase in lifespan. So we're pretty sure of the pathway that we're activating here. And when we looked at gene expression, it's kind of amazing that just eating less changes about 8% of the expression of your genes. Okay, so about 17,600, yeah, that number. Out of about 22,000 genes. I mean, that's a lot of genes to change just by eating less. That's not like eating differently. It's not eating some strange food, it's just eating less. And then oxaloacetate produced other gene expression change of about a quarter of them, of these, were similar. And of the ones that were similar, the genes were either expressing more proteins in unison, or they were expressing less proteins in unison. They weren't doing this, okay? So they were mimicking at least a portion of calorie restriction. Now, is that enough? Are there enough genes copied to get the full benefit of calorie restriction? We don't know, so we test. And just so you know some of the genes, for instance, here's the FOXO3A gene, which deals with glucose homeostasis, regulation of transcription, and response to starvation, i.e. calorie restriction. In calorie restriction, we see a 100% increase of the activity of this gene as compared to a control group that's fed normally. And when you supplement with oxaloacetate, you get about a 70% increase. Pretty impressive. Okay? Same with some of these other genes. And there are another 300 and so that are doing the same thing. So let's look at some of the applications of oxaloacetate that we've been able to verify. And just as a review, here's the Krebs cycle. Here's oxaloacetate. It goes into citrate. If it goes backwards, which is what we're doing, it goes to malate. Okay? And the effect on, we started off with very simple animals. So we started off with C. elegans. They live a short time. So here's 100% alive. Here's 100% dead. And what you want to do is try to move this curve to the right as far as possible. So here's the control group. And you can see when we put in different concentrations of oxaloacetate, it not only increases average lifespan at 50% survival, but it increases maximal lifespan. We see a 25% boost in these little animals living longer just by adding oxaloacetate to their agar. They live on little plates. Now, when someone else in the community has repeated this experiment, they just published this 2 months ago, instead of seeing a 25% increase in lifespan with oxaloacetate, they saw a 49% increase. They had to up me one, which they did. When we did this in mice, we saw a 25% increase and an increase in maximal lifespan also. We did this in flies, and we saw the same thing. So essentially, we're seeing about a 25%, in our experiments anyway, in 3 different species now. And this is very similar to what we're seeing in calorie restriction. We're seeing very different species reacting through this pathway to increase lifespan. And they're not just living longer. The mice had less graying of the fur. They had less curvature of the spine. They had less inflammation. We could tell that because they itched less. They were more active. So the goal is not to live longer here. The goal is to live longer and live better. They did a clinical trial of oxaloacetate in Japan in 1968 in diabetics. What they found is summarized on this chart. The red bars indicate the amount of reduction in fasting glucose levels. So the first thing you notice is that every patient had a reduction in fasting glucose level. The second thing you'll notice is that the average across the board is about 25%. There were no side effects. They were looking at both type 2 and type 1 diabetics, which is really interesting. Have you ever heard of this study? It was in the Tohoku Journal of Experimental Medicine. I looked for the follow-on study. I mean, here you've got a natural human metabolite that's lowering fasting glucose levels in diabetics by 25% in less than 40 days with no side effects. Where's the follow-on study? There was nothing.
Where's the drug?
Where's the drug? There you go. I went to Japan. It turns out the University of Sendai is right up by a place called Fukushima. Yeah, that one. And I found the department that dealt with this, and I said, "Are you familiar with this study?" And they said, "Yes, we're very familiar." I said, "Where's the follow-on work?" And they said, There was no follow-on work. I said, why not? They said, well, it's a natural compound. I said, yes! I said, it's a human metabolite. There is no toxicity, at least none that we've been able to find, and we've done the tests. We did the GMP toxicity tests, okay? And they said, yes, natural compound, no patent. And that was the end of the conversation. So, but more interestingly, from a nutritional supplement side, because we can't talk about diabetes, and we can't sell to diabetes, but we can talk about glucose stabilization in normal people. So here is a somewhat normal person, and you can see, even though he's in the range of being normal, his fasting glucose levels are all over the place. And this is just fasting glucose levels. His glucose levels throughout the day are all over the place too. After he starts taking oxaloacetate, after about a 3-week period, because this is a genomic retooling as opposed to a pharmacological retooling, you see about an 8% drop in glucose to the low end of normal, but look at how tight his levels are. This is a 55% drop in the swing of glucose, and it's because of that upregulation of that FOXO3A gene, which deals with glucose homeostasis. So is it important in normal people to maintain a stable glucose level? Yes, because most of the damage occurs when things swing up, and most of the problems occur when things swing down. Okay? Here's a study that was done outside of this country, because we can't do studies in this country. This is a 73-year-old European woman. She was on 3 anti-diabetic medicines, and she started taking oxaloacetate. And we have her levels at fasting, which is the light blue. Red is breakfast, black at bedtime. And you can see the swing she has in her glucose levels throughout the day at the start of the study. This is 0 to, it went 75 days. Okay? She's all over the place. She's on 3 anti-diabetic medicines, and she's all over the place. After taking oxaloacetate for 75 days, look at how tight her levels are. Look at her control. Okay? So, as a summary of what we found with her, her fasting glucose levels dropped 23% to normal. Glucose levels after a meal dropped 34.5%. You know, you've heard of the glycemic index? You can do it 2 ways. You can either eat low glycemic foods, or you can get your system working properly. It's your choice. Her metformin use during the study dropped 50%. And incidentally, metformin, the mode of action is activation of AMPK. Isn't that interesting? Okay, and the results are statistically significant here. So one of the other things we've seen is a reduction in fat mass. If your body thinks you're eating less food, you're going to generate less fat. Okay? It actually turns down the genes that create and store fat by about 20 to 30%. And when we measure this with nuclear magnetic resonance, we see the fat mass is dropped not as much as every other day calorie restriction, but a significant amount. In this case, it was about 29%. And interestingly, muscle mass is not significantly reduced or free fluid. Now, do any of your patients have a little more fat than they want? I do. What we found is this is not really so good as a fat loss compound, but rather after you go on a diet, you'll drop in weight, and then you do something called rebound back. And there are biochemical reasons why you rebound back. One of the reasons is you turn on genes that help you store energy because, hey, we're energy low, we need this energy. Store it. We don't know when we might need it again. So what this does is it slows down the rate of rebound. You'll see that on the next slide. So when we start giving oxaloacetate to animals, they still gain weight. They just gain it at a slower rate. So think of this as an after diet compound as opposed to a diet compound. How many of you are dealing with weight loss where your patients lose 30 pounds in a month, and then the next month they gained 32 pounds? Congratulations. If you can help them to gain that 30 pounds back over 2 years, think how much more successful their diets will be. Think how much more successful their health will be. So, some other interesting things as a nutritional supplement. Oxaloacetate protects mitochondrial DNA in the brain. Is this like the Mecca? What they did is they took cyanic acid, which is known to destroy mitochondrial DNA and cause seizures. They injected it into rats. They then gave the rats intraperitoneal injections of oxaloacetate. They had to do this within 2 hours of the initial injection. And there was no DNA damage. That's pretty cool. Okay? Retinal pigmented epithelium cells damaged are protected by zinc and oxaloacetate. Okay, that's an interesting thing. Pancreatic islet cells and neurons are protected by oxaloacetate. Isn't that interesting? And it gets even more interesting because in the last 2 years, they found that an enzyme called pyruvate carboxylase in diabetics is down-regulated. Its activity is reduced in type 2 diabetics. Okay? And when we take that enzyme and we overproduce it in animals, we can't get them into a diabetic state. When we get rid of that enzyme in the pancreas in animals, they almost immediately go into a diabetic state. The function of that enzyme is to take pyruvate and make oxaloacetate. Okay? When we give oxaloacetate to diabetic animals, it proliferates the beta cells in the pancreas that make insulin. Isn't that interesting? Okay, and lastly, oxaloacetate is a powerful antioxidant. It drops T-bars production in the brain. It does all sorts of neat things. I'm not sure that its activity is due to its antioxidant qualities, but certainly it's not a bad thing. We also see an increase in endurance with oxaloacetate. We took individual muscle cells and we mounted them on transducer plates. And then we did the old frog legs experiment with electricity. And what we saw is that we could drive these muscle cells into fatigue and measure the amount of time it took to go into fatigue. We then used physiological levels of oxaloacetate in the solution, and those same muscles took 10% longer to go in fatigue. Do any of you have any patients that suffer from fatigue? One. Okay, good. This is a healthy group. Okay, so we not only see this as a possibility, and this was presented at the experimental biology meeting last year in Washington, D.C. Not only does this have application for your elderly patients, but also for the weekend warriors Who wouldn't want a little more endurance? It doesn't give you any more strength. Okay, so you can't all of a sudden lift 10% more. But you can continue doing the things you love for 10% longer. So, as it turns out, we have some national medal-winning triathletes that take oxaloacetate before every match. And I asked them, I said, will you tell this to the world? And they said, no! Are you out of your mind? No, it's just a thought. So let's talk, now I've got to separate this completely, and I've got to make sure that you understand. I've been talking about ways to use oxaloacetate as a nutritional supplement. We do not diagnose, treat, cure, or prevent any disease. Okay? Now, let's talk about the science of disease over here. Okay? It's important that there's a separation here. And it's important to know I'm not suggesting you use oxaloacetate for any disease you see up here, other than through standard clinical trials. And I hope you join with me in some of those trials. So, potential support for cancer, Alzheimer's, Parkinson's, stroke, epilepsy, diabetes. There are companies that are bringing all of these into clinical trial this year or started last year for oxaloacetate supplementation. Okay, the animal data looks good, and again, oxaloacetate is not intended to diagnose, treat, cure, or prevent any disease. And I do mean that. One of the things we've seen with calorie restriction is it decreases cancer risk. Well, that's interesting because when we give oxaloacetate to human lung cancer cells, they stop reproducing. Well, that's interesting. The neat thing is oxaloacetate really has no side effects on the normal cells. They eat it up as food. What's happening in the cancer cells is they are so dependent on glucose that they store the oxaloacetate in junky bodies. These junky bodies get bigger and bigger and bigger, and it interferes with mitosis, and the cells stop dividing. It's kind of a simple mechanism. And that was published in 2007, and you've all heard about that, right? No. Yeah, okay. Okay. Oxaloacetate reduces glioblastoma, one of the worst brain cancers out there. Survival rate, 5-year survival rate of 2% in humans. Oxaloacetate reduces tumor growth by 50%. This is done, what they do is they take nude mice, because they die of everything, and we implant human glioblastoma tissue into the mice, and then we see how long they live, which is the control group, and then we give oxaloacetate in the water to the other mice, and look at the difference. Wow, that's pretty cool. But it gets better. Here's a mouse without anything. You can see the tumor there. That mouse is toast. If you include oxaloacetate in the water, it's about 50% less. So this is control oxaloacetate. This is a chemotherapy drug, temozolomide. And note the change in scale here. Okay, so chemo works a lot better than just oxaloacetate alone as far as the size of the tumor. But if you combine them together, look at this. Look at this. That versus this. Look at this. Synergy. Synergy is good, especially if you have glioblastoma. And what's interesting as far as survival is the combination increases survival 237%. A third of the mice, we couldn't find the cancer cells. Here, we put the cancer cells in. I know they were there. temozolomide and oxaloacetate, a third of the mice, we couldn't find the cancer cells after about 2 months. Is that a good thing? Yeah, probably. Well, the FDA thought it was a good thing. So they issued us an orphan drug status for the treatment of gliomas for oxaloacetate, and we're going into clinical trial this year for glioblastoma. Now, the good news here for those people is no side effects. So quality of life is not impacted by this. This is a good thing, right? And it was the combination of oxaloacetate and temozolomide that showed a third of those animals were in remission, okay? But in the human case, we actually cut out the brain cancer. We also give radiation. We also give chemotherapy, and now also oxaloacetate. And one of the reasons oxaloacetate works so well, we think, is because in December of this last year, it was shown that AMPK activation actually allowed cancerous stem cells to start differentiating normally and produce normal cells. So we're not just talking about it going away for a while. We're talking about changing the metabolic environment so that it doesn't come back. So it doesn't come back. Okay, let's talk about something other than cancer, Parkinson's disease. I got a call from one of my clients and one of my customers, and they said, oh, you got to send me a bunch of oxaloacetate. And I said, sure, okay. Why? Well, I've got Parkinson's disease. I said, yeah, okay, why? They said, well, my neurologist suggested it. I said, okay, that's interesting. Increased mitochondrial function. I can think of a couple reasons why it might work. Well, this is her the quality of life survey that she did, and the red lines indicate how Parkinson's affects your life. So the bigger the line, the more it affects your life. This woman was in a wheelchair. She couldn't comb her hair. She had trouble feeding herself. She couldn't button her buttons. And most importantly to her, because she was a professor of pharmacology at the University of Kansas, she couldn't type and type up research papers. It was really bothering her. She started taking oxaloacetate. She had a 90% reduction in symptoms. She went from a wheelchair to total functionality, and she took a trip to Europe with her husband. Has her life changed? She really likes me. So we thought, well, let's take a look at some other people. And what we did is we took some people who were Parkinson's patients who were interested in oxaloacetate for anti-aging, because we can't treat disease, and we gave them a self-study, a self-filling out study, a PDQ form, PDQ 39, which is a validated study, and we looked at how effective it was. And what we saw is it worked in about half the patients. Half of them it didn't do anything to, plus or minus. The other half, they had some positive effect. And the average positive effect was 63% improvement. And that's statistically significant. And the range varied anywhere from a low of 20 to 90%. So based on this, the University of Kansas said, we're doing a clinical trial. We love this stuff. Our professor over here is like poster child. So this is now in clinical trial. It's a double-blind, placebo-controlled trial at the Parkinson's Disease and Movement Center at the University of Kansas. And you can follow along with our progress here at clinicaltrials.gov. Let's talk about Alzheimer's. We've been working with the Pennington Biomedical Research Facility in Baton Rouge, Louisiana. I believe it's in Baton Rouge. Might be somewhere else in Louisiana. And what they saw is they looked at a control group of Alzheimer model mice. So how closely those mice model Alzheimer's, I don't know, but they say they do. And they looked at every other day feeding. They looked at another compound that's another calorie restriction called restriction mimetic called manoheptulose. I believe I pronounced it. It's a sugar from avocados. Interesting. And oxaloacetate. And what they found is the secreted amount of this APP alpha protective compound was elevated in calorie restriction, the MH, and the oxaloacetate material. So that's good news. What's also good news is we dropped intercellular A-beta proteins, 1 through 40, 1 through 42. And really interesting is we dropped hippocampal plaque density with oxaloacetate and every other day feeding, tying into the fact that calorie restriction helps Alzheimer's. When we put these animals in a water maze, we saw a 50% reduction in short-term memory loss with oxaloacetate. Do any of your patients have memory loss? Where are my keys? So let's go to the fourth part of this, patient profiles. As a nutritional supplement, we have to stay away from the disease, and we want to do this legitimately. We want to be above board because we've got a lot at stake here. Okay, we've got something that may be able to help a lot of people. And so as a nutritional supplement, we can look at anti-aging, general. I mean, anything that increased lifespan in 3 animals, 3 different types of species, might be good. Helps to maintain proper glucose function, so supports proper glucose functioning. Weight maintenance, reduces the rebound effect. Increases endurance, fights fatigue. So your elderly patients, fatigue patients, they don't have to be elderly to be fatigued. Your athletes. Some other funny things we found, mood swings in PMS. It turns out that during PMS, there's a spike in glutamate levels in the brain, at least in our animal models. And when we give oxaloacetate, it combines with the glutamate through the enzyme serum glutamate oxaloacetate transaminase, or SGOT, into alpha-ketoglutarate, which instead of being a brain excitatory chemical, is a brain protective chemical. And women who have secluded themselves for 2 days a month, every month, since they were 13 years old, all of a sudden don't see the mood swings. We've tried this in 12 patients. 11 of the 12 responded favorably, with anywhere from a 25% reduction in mood swings to 100% reduction. We've changed these women's lives. Imagine if you don't have to miss 2 days of work every month. And all with a nutritional supplement for a non-disease state. Constipation reduction. It turns out oxaloacetate is a driver of the lower intestine, so just giving some of this helps a lot of people with constipation. And improves mitochondrial function. It turns out that AMPK activation is one of the first steps in generating more mitochondria. So we haven't published this yet, but we've actually been able to see higher densities of mitochondria in the brain after oxaloacetate supplementation in animals. We haven't cut up people yet. So Now, as an experimental clinical trial, help for Parkinson's disease, glioblastoma, Alzheimer's disease, 2-hydroxyglutaric aciduria. We're actually in clinical trial with this right now. This is a phase 1 trial at the University of California, San Diego. And mitochondrial diseases, diabetes. So what do we need for a clinical trial? Because I want to enlist you. I want to enlist you to help me. Okay? This is a group that thinks outside of the box. As a matter of fact, I don't know that you've even found the box. Okay? Could oxaloacetate and ozone be effective for various conditions? Well, if it's a non-disease condition, that's easy. You just try it. If it's a disease condition, you can still try it. There are only 3 things that you need. Well, 4. You need IRB approval. And if you have trouble with that, I can help you with that. There are private IRBs that look at your plan and give you some feedback and say, you know, hey, I wouldn't give them 6,000 capsules of this. I'd only give them 3,000 capsules. Okay. Data sent to clinicaltrials.gov. That's required by law. Okay, so you're going to be reporting to the FDA, and I know that scares some of you in this room. It scared the heck out of me. You need a patient approval form, and you need to give FDA 30 days pre-notification to the trial. So you put together a book. I've got all the information that you need to give to FDA. What I need from you is a protocol that makes sense based upon your own knowledge. Okay? And if you get that, then we can do a clinical trial legally in these patients, and maybe we can help wipe out some of the diseases here and make people a little bit better. Now, over here, you'll see, okay, plug time, promotion time. Over here, I've got some bottles of Benagine, which is our version of oxaloacetate. It happens to be stabilized, so it's got a 2-year shelf life, which was probably the hardest thing in bringing this as a nutritional supplement, because when we first started working with it, it had about a 2-week shelf life. And I've got some DVDs, if you'd like to see more on this, and I've got some brochures and a business card. So during the break, you can come down, just grab some. I don't have enough for everyone on the bottles, but if you grab my business card and email me, I'll send you a free bottle. No big deal, right? So if you believe in what we're doing and what we're trying to do, help me form a clinical trial so we can test it out. Test out some of your ideas. Or use our nutritional supplement, because we use the monies that we gain on this, that we get on this, for doing other research. I have no external research. I did all this research with my own money because it's something I believe in. This is why you became doctors, because it's something you believe in. So let's change the world.
We have a little time. All the people that are aging really fast better get that. The others of us who are aging more gracefully, we can just sit in our chairs. Alan, we have a little time for some questions.
Sure. Thank you so much for your presentation. What's the dose? What's the cost? And do you have to stay on it long term?
Well, first of all, can you still hear me? I don't think you can. Hold on. Okay, the question was dose, cost, and how long do you have to stay on it? The dose depends upon what you're trying to do. We found that for general anti-aging, we're able to activate AMPK with 1 capsule a day, which is 100 milligrams. For glucose stability, you're looking at probably 2 capsules a day. I usually do it in the morning and in the evening with a meal. For some of these other things, like H2 hydroglutarate aciduria, we're using up to 6 grams a day, which is equivalent to 60 capsules a day in infants. Okay, and we're having some success. So it depends on what you're doing. As far as how long do you have to be on it, obviously if you have a genetic mutation and you're missing a gene that is doing an important function, I can't put that gene back into you. So you'd need it for the rest of your life. If you, for instance, with glucose control, it may be that if you bring down your glucose levels and get your pyruvate carboxylase functioning again, you may be able to discontinue it. But we're still early in trying to figure that out.
Have you tried calorie restriction and supplementation?
I've tried calorie restriction, but I haven't tried it with supplementation. And I often get asked, will it help people who are already doing calorie restriction? And my response there is, well, how thin do you cut your cheese? Think about that one.
That was a great presentation. As an oncologist, I'm a radiation oncologist. I'm very interested in becoming part of this project. Absolutely. I know I wouldn't make it to the box, so
okay.
I decided to grab the microphone. But if I use a lot of high-dose intravenous vitamin C, hydrogen peroxide during radiation, chemotherapy, surgery, or as a sole treatment, how would that interfere or help with the oxaloacetate?
Well, I don't know what you're using, and there are a zillion different drugs out there, so I don't know the interactions with all of them yet. So far, we haven't found a negative interaction with oxaloacetate in any drug, and that's not that surprising because oxaloacetate is a key part of our metabolism. If there was a drug that negatively interacted with it, you die, and it would never make it into clinical trial as far as the other drug. So we think it's safe.
Thank you.
Question, just, I guess the whole scenario where the government is trying to take supplements and make them drugs and control the access to supplements as, you know, put it under the doctors who know everything about supplements, right? Ha ha. You know, could this orphan drug status then be used against us to use this as a supplement and keep it free?
We've been really careful. If you apply as a drug, even as an orphan drug, with a nutritional compound, then FDA will come back and say, well, this is a drug and it cannot be used as a nutritional supplement. We were careful enough to establish a market as a nutritional supplement first and did all the paperwork under DSHEA for a new dietary ingredient. We spent $1 million on proving low toxicity through GLP trials so that we could be a nutritional supplement. So if you do that first, then they can't take it away from you. Now, I can't go and start selling this for cancer or Parkinson's or diabetes or any other drug or any other disease unless I have FDA approval. But we can still use it as a nutritional supplement for non-disease states. And there are enough of those that I'm sure you can find at least one.
Does this compound have the same effect long-term or does the body kind of figure out it's not really on calorie restriction and compensates and it doesn't have the same effect?
We've actually done studies for the entire lifespan of mice, and we're seeing the kick all the way. Now, there have been 3 studies in mice. One was done by the National Institute on Aging, one that was done by us, and one that was done at UC Riverside by Steve Spindler. And we saw different results in all 3 studies. Imagine that. It's like science. We saw the increase in lifespan, but we were using stabilized oxaloacetate. The NIA saw no increase in lifespan, no decrease in lifespan, but when I got a sample of their chow that they were using with the oxaloacetate in it and sent it to a third-party lab, the amount of oxaloacetate in it was 0, non-detectable. So essentially they had a control group, a second control group. The third study, which was done by Steve Spindler, saw an increase in lifespan of about 10 to 15% for 90% of the mice's lifespan, and the last 3 mice died before the last 3 mice of the control group. And we don't know why. So a little bit of mixed results, which is not uncommon in complex biological systems.